All right. Our subject for this week, we're still talking about heredity and how things get passed down from parent to offspring. We said Gregor Mendel was the guy who worked with pea plants. He figured out that things get passed down, but didn't really know how they got passed down, like the science behind it. And we didn't really discover that until we learned about DNA. DNA has always been there, but it's not always been known what it was. In fact, it wasn't discovered until 1950, which I know that you guys think that 1950 was a million years ago. It's not that long ago in the grand scheme of things. In fact, my parents are born in 1949, so they didn't really learn about DNA in high school because it was too of a new subject. So it's not been around that long. So let's get into what exactly it is. So DNA is kind of like the blueprint for your body. It's a set of instructions that is unique to every single individual. Unless maybe you're an identical twin, you might have the same DNA then. But for the rest of us, it is our own unique molecule that's inside the nucleus of our cells that tells our body what to do and how to do it. So let's do a little history lesson here, and then we'll get started. So DNA stands for deoxyribonucleic acid, or DNA for short. It's your genetic material, and it's your basic set of instructions. A little history, we have to talk about a couple scientists here. First one we're going to talk about, is Rosalind Franklin and Maurice Wilkins. They worked in X-ray crystallography, which basically they're taking X-ray pictures, and they're studying. They're trying to discover DNA and what it is and what's inside our cells that makes us who we are. And they are famous for this picture right here, which is called Photograph 51. It's an X-ray picture of DNA, and you can kind of see like the double helix here if you look at it from like above. And they were kind of racing to be the first ones to discover what DNA was and what it looked like and what the structure of it was. There's some controversy because, oh my gosh, my cat is up here. Get down. I'm doing something. Go away. So there's some controversy here because when you think of DNA, they got a hold of this picture right here that Rosalind Franklin took and Maurice Wilkins took. Somebody leaked that picture to them, that X-ray picture, and it gave them the information that they needed to go ahead. Think of these two guys here. You think of James Watson and Francis Crick. James Watson and Francis Crick were the first two that actually came up with the structure of DNA. They built the first model of it called the double helix in 1953. And the controversy is that they almost had their model done, but they were missing something. They didn't have what they needed to kind of put it all together. And the controversy is that and finish their DNA model called the double helix. When you think of DNA and you think of the people that get the credit for it, you think of Watson and Crick. You don't think of Rosalind Franklin very much, which, in my opinion, she kind of gets, I don't know, she doesn't get the credit that she deserves. In fact, Rosalind goes on, she lives until she's 37 years old and she dies of ovarian cancer, and many believe that the x-rays that she used to take pictures of DNA molecules might have caused her to get that. Cancer. So Watson, he's still alive, by the way. He's 92 years old. He is this dude right here down on the corner. He has an Indiana connection. He got his PhD from IU back in 1950, so he's a Hoosier. Then he went on to England and studied over there, and that's where he made his DNA discoveries at a college in England. Francis Crick, this dude here, he's dead. He died in 2004. So they get the credit. She doesn't really get a whole lot of credit. And they got those guys plus Wilkins on the first page. They go on to win the 1962 Nobel Prize in Medicine, which is a big deal. Um, Franklin doesn't get it because she died in 1958. So Watson and Crick kind of argued like she should get it too, but the Nobel Prize people said, "Nah, we only give our awards to living people. We don't give it posthumously." So she kind of gets shut out. So it's kind of a shame. She deserves the credit. All right, so let's look at their discovery here and what DNA is actually made up of. Alrighty, so it is a long, twisted molecule that they call a double helix. It's two-stranded. It is made up of these structures called nucleotides. So here's a nucleotide. Nucleotides have three parts to them. They have a part called a phosphate group. They have a pentose sugar. Pentose means it's like a five-sided. So it's a pentose sugar, or to say sugar, phosphate, sugar, and an organic base. Or you can call them a nitrogenous base. There are 4 bases to choose from, and they are down here. The 4 bases are called adenine, thymine, guanine, and cytosine. I'll talk about them here in a second. The way that these little nucleotides pair up, and there's one, and here's one over here. They form the 2 sides. So you've got sugar phosphates on the outside, and you've got on the both sides on the outside of your sugar phosphate groups, and on the inside are your little bases, your A, T, C, and G. They're called complementary bases because they go together in a certain way. They complement each other. So, for example, A always pairs with thymine. So adenine and thymine always pair up. And guanine and cytosine always pair up. So they go together. So complementary, I know it can mean like, oh, you look nice today. That's a compliment. But in this case, it means like goes together. Like, I don't know, your foot and a sock. They complement each other. They go together. I don't know. Well, ketchup and mustard goes together. I can't think of anything off the top of my head. Peanut butter and jelly goes together. So those are complementary. And so they hook together and they make up what you would call the rungs of the ladder. So if you had a big gigantic step ladder, where you put your feet, those are the rungs. So these are the rungs here. They make up the inside of DNA. And on the outside is your sugar phosphate groups that makes up the legs of the ladder. So there's a leg and there's a leg. And you can see it makes a code here that you can read. So if you read it from top to bottom, it goes AGTZ. That code, I mean, think about it. You've got 4 different bases. And if you had a code of DNA, I don't know, let's say it's 1,000 nucleotides long. I mean, how many times could you rearrange those letters and get a slightly different DNA strand? I mean, a ton. You could keep changing the code and changing the code and changing the code, and it would be like impossible to come up with the same code. That's what makes us all unique and all different. My code of letters, my sequence of letters is different than your sequence of letters. Everybody's sequence of letters is different except for identical twins. And even at that, I don't know if it's exactly perfect, but it's pretty close. So that's what makes DNA. When you watch all these crime shows and people, they commit crimes and they find like blood, or they find skin cells maybe, or they find a couple hairs of the suspect. They can run a DNA analysis on it, and if your DNA is on file or on record because you've committed a crime before or something, or that you're a suspect, so they take a sample, it is really, really, really good data. Once they find your DNA, nobody else has the same. So that's how they put people in jail for crimes, because they can prove it. It was you if you left something behind like that. So what I want you to do right here is I want you to pause this video, or just remember where you're at. You're about the 7-minute mark. And I want you to go back into Google Classroom and find a video called, I think it's DNA structure is what I called it. So I have a model of DNA that I'm going to show you in this video and talk about it. So pause, go watch that, come back. Thank you. Another thing that we have to talk about that DNA does is it replicates itself. And replicate means make another copy. So if you're going to make a replica of, I don't know, the Eiffel Tower out of popsicle sticks, you would build a model of it. It's much smaller. So you build a copy. So that's what replication means, to make a copy. Why would DNA need to make a copy of itself? Well, think about it. 13, 14 years ago, you started out as one cell. So now you are made of trillions of cells, and all those cells have to have DNA inside the nucleus of their cells. So there had to be a way, or there has to be a way, that your DNA can make copies of itself, so that every brand new cell in your body also has a set of instructions that tells the cell what to do. So there has to be a copying process. Also, your cells don't live forever. They die off and they are replaced with new cells that take their place. You know, think about your skin cells. Your skin flakes off all the time. You replace it with new. If we didn't make new cells, we would never be able to heal bones, heal cuts and scrapes, grow. Your fingernails wouldn't grow. Your hair wouldn't grow. Your body wouldn't grow. So mine's growing outward right now instead of upward. So anyway, we've got, because I'm getting fat. So we've got cell growth happens all the time. We're making new cells. So they all have to have DNA. So how does this happen? How does DNA make another copy? So I try to put a picture on here that helps show that. I also made another video that you'll pause here in a second and go watch it and come back. So first thing that happens is DNA, since it's tightly wound in this double helix structure, it's got to unzip itself. So the way it does this, first of all, it flattens out and it untwists, and you can kind of see that process happening here where it's getting flattening out. Next, it sends a enzyme down it. This is that yellow thing here. You don't need to know its name. It's just an enzyme. Its job is to unzip. Oops, I circled the wrong one. That's the unzipper right there. So he unzips the DNA, which exposes the bases, the ATCG, and then they go find nucleotides floating around in the cell, and they bring them in, and they read the code, and the appropriate letters match up. So if you have an exposed thymine, and an adenine will pair up with it. If you have an exposed cytosine, a guanine will pair up with it. So you can see these little guys right here. They're coming in, they're reading the code, they're pairing up, and it's creating another new strand. So it uses half of the old strand kind of as like a template or a model so that it knows what to do. So when it's done, it's created this new strand of DNA, and it'll do the same thing down here, creates a new strand. And the new strands of DNA, it's like half of it's old and half of it's new, because this is the old piece here, and the new piece is going to form on the other side. So it's like half old, half new. Half of the strand came from the original DNA and half of it didn't. And then when it's all done, it coils itself back up into the double helix, and now you have two where you used to have one. And then the brand new cell that's formed gets one of those strands. So that's how it works. This is called DNA replication. I want you to, well, you know what? Don't do that yet. Let me finish one more thing, and then you'll go watch this video. All right. Let's skip ahead here to the next slide. The next slide is talking about, well, what would happen if when we're making a copy of DNA, if we're replicating DNA, what would happen if this copying process got messed up? Something happened, something went wrong. This is called a genetic mutation. A genetic mutation is a mistake in the nucleotide sequence for a DNA strand. Mutations happen during replication all the time. Fortunately, your body kind of proofreads the DNA after it's done copying itself, and it looks for mistakes. And if it finds mistakes, then it goes in and it cuts out the bad part and replaces it and makes it the correct code. And hopefully your body does a really good job at this and keeps up with that. Because if it doesn't, then it leads to certain kind of conditions, and this is where cancer might come in, because cancer is basically a genetic mutation. All right, here we go. There's three kinds of genetic mutations, something that goes wrong in the copying process. So you want to look at this example right here. So in this example, it says your normal DNA strand is going to have this code, GGCCTT. All right, so if you look at the green right here, so instead of GGCCTT, it goes GGCATT. That A is not supposed to be there. It's supposed to be a C. So what has happened is the letter A, adenine, substituted itself for the C that's supposed to be there. This is called a substitution mutation, where one base letter is different than the other one. So that's substitution. Second one down the row here. Now we go back to the normal DNA strand at the top. It's supposed to be GGCCTT, and it's not. It's GG, that's fine. CC, that's fine. A, where the heck did that come from? And then TT. So that A in the middle there, that's not supposed to be there. That's an extra letter. So that is called an insertion. I've also seen people call it an addition, which is same. So that's an addition mutation. That shifts the code down. Then I can do this one. And that bottom one, it's the GG, that matches with the normal DNA. C, it's missing a C. So there's a C missing. It's been deleted, so it's gone. And that also makes the code shift. The easiest thing to do on these, and you're probably going to have a question about this, I got to make the worksheet here in a second, is if you know what the normal strand is up here, you just count the letters. It has 1, 2, GG, 1, 2, CC, 3, 4, TT, 5, 6. That's 6 letters. Then if you go count these, letter A has 1, 2, 3, 4, 5, 6, also has 6. So that screams substitution to me, because substitution doesn't change the amount of letters, doesn't the amount of bases. B has GG, that's 2, CC, that's 2, that's 4. A, that's 5, TT, 6, 7. That's 7 bases, so that has an extra base. So that should scream to me, that's an insertion, that's like got extra stuff. And then C has GG, C, TT, that's 1, 2, 3, 4, 5. That screams to me, that's one base, not enough. So that's got to be a deletion. All right, so those are the three kinds of genetic mutations. Next topic. Mutagens. Actually, we're not going to do this topic here, so scratch it out. Mutagens. You could also call them carcinogens. Carcinogens and mutagens are anything that can cause your DNA to have a higher likelihood of having a genetic mutation. So things that you don't want to do, because they would increase your chances of having genetic mutations and would increase your chances of having cancer. Put them here at the bottom, some examples. So UV light from the sun. Everybody knows that you shouldn't go outside, lay in the sun for 8 hours a day and just get baked by the sun, because that increases your odds of having skin cancer. So if you go outside for an extended period of time, you should put on sunscreen, especially me on the top of my head, or I have to wear a hat, or I will burn my head with UV sunlight. And that could be harmful, so that could cause cancers. That's a mutagen. X-rays, obviously we have to get them for our teeth, probably once a year from the dentist. If you ever injure yourself, you might get an X-ray. They don't last very long, just a couple seconds. But if you did them over and over and over and over again, they are risky. You do they usually put like a lead vest or something on your chest if they're taking an X-ray of your teeth to try to protect your internal organs. But there is some risk with X-rays because it's got radiation in it. Certain chemicals, cigarettes, tobacco products have certain chemicals in them that might that cause cancer. Um, they say processed foods and food preservatives, which we eat a lot of that as Americans, processed foods and preservatives. That could be cancer causing. Certain chemicals and cosmetics and cleaning products, you might be careful about using them. Um, let's see. Viruses, like the human papillomavirus, the certain bacteria might cause it as well. So there's certain things. There's a chemical called asbestos that is used as a uh, it's used to coat pipes and things in buildings. It's like a trying to think here. Got to get my terminology right. It's like an insulation kind of material, and they don't use it anymore really in buildings. It's in old buildings they might have it. In fact, our school, they're currently working on the cafeteria in the high school. The high school's a pretty old building. The middle school is the newest building. It was built in I think 1997, so it's not that old. It doesn't have any asbestos in it, but the high school did. And over Christmas break, when they were getting ready to work on the cafeteria, they had to go in and remove it. And what's bad about asbestos is when you try to remove it, it breaks up into little bitty fibers, little bitty small particles. They can get into your lungs and causes a condition called mesothelioma. I don't know if you ever watch commercials where they jump on and these lawyers are like, have you ever, somebody you loved has mesothelioma? Call this hotline number. Well, that's what it is. You breathe asbestos, and you can get a certain kind of lung cancer from it. The school removed it. You have to have a team of people come in, put up plastic tarps everywhere to seal the room off, put on protective clothing and respirators, and take it down and seal it up in bags and remove it so it doesn't contaminate anything. So it's been done. It's gone. So anyway, um, now what I want you to do is I want you to go and watch another video I created called DNA replications slash mutations, which will kind of help go over this material again. So um, that is it for this week. I'm going to post a worksheet that goes along with this.